So let's continue with the biochemistry, and in the previous video we discussed about the amino acid and we are continuing with the amino acids. Kwa sasa hivi naangalia properties. Amino acids kwenye property kwanza, all are white crystalline. Kwa hiyo ukipata amino acid moja inakuwa white crystalline, lakini huwezi kuiona amino acid moja because most of time tunazipata zikiwa kwenye protein na zikiwa tayari complex. Kwa ndio maana unaiona protein kama protein. Lakini pili, they are amphoteric substance having basic property by virtue of amino group and acidic property due to carboxylic group, kama ambavyo nimeelezea kwenye previous lecture kwamba tukiona structure for amino acids like the amino acid like this, then kuna basic group, kuna acidic group, so they are amphoteric in nature. And then we have the study question here. Let's solve this first, then we will continue with the properties. So sometimes teacher, they can ask you what structural features account for the amphoteric property of amino acid. Kwa hiyo kwenye kujibu naambia tu kwanza consider the structure of amino acid. Ukisha draw this structure of amino acid, then what you are coming to say that we have the amino group here and we have the carboxylic group, and you are labeling this diagram. Then you are saying that the amphoteric property means as both acidic and basic properties. The acidic property of amino acid is due to acidic group, and the basic property of amino acid is provided by the basic group. Now the name amino acid is due to amino group, and the name acid is due to the acidic group. So sometimes in your explanation you may include maybe the acidic group. The acidic property of amino acid is due to acidic group. You may put in bracket here and you may write the acidic group. And the basic property of amino acid is provided by the basic group. You may put in bracket here. And then you may write NH2. The third property, they ionize in aqueous solution. Those with one amino group and one carboxyl group will give neutral solution. Those with more carboxyl group than amino group will form acidic solution. And those with more amino group than carboxyl group will give basic solution. Kwa hiyo unaweza kufikiri kwamba kama tumesema amino acid ina amino group na ina carboxyl group, wanasemaje kwamba with more? Sometimes kwenye ile group ambayo ni R, ile group unaweza kukuta na carboxyl group nyingine au ina amino group nyingine. Kwa hiyo pale kwenye R ndo panapoleta variation. Note, amino acids are soluble in water where they form ions. These ions are formed by the loss of hydrogen atom from carboxyl group, making it negatively charged. This hydrogen atom associates with the amino group, making it positively charged. So in a solution, the amino acids, they become charged because of the dissociation. Now let's again come back to the structure of amino acid, which is carbon, then R, hydrogen, then carbon, double bond oxygen, then hydroxyl, and then here we have nitrogen, and then you have hydrogen, and then hydrogen. So here, if you dissolve this in water, what will happen is that the hydrogen from carboxylic group will be released and will go to associate with the nitrogen of the amino group. So what will happen here, it will be carbon, then R, then hydrogen, then carbon, double bond oxygen, then O. Now this will become negatively charged, and then you will have nitrogen, then hydrogen, then hydrogen. So the hydrogen from this oxygen is associated with the nitrogen, and we may draw this arrow showing that this hydrogen has moved from here to here. So what happens, this will become positively charged and this will become negatively charged. This will become positively charged because of the addition of hydrogen, and the addition of hydrogen here makes this to be positive. So if amino acid has only one amino group and only one carboxylic group, means this negative charge will cancel with this positive charge and the whole molecule will remain neutral. So this ion formed here is what we call it as Zwitter ion. So this is called a Zwitter ion. So what you can say that molecules like amino acids which contain both acidic and basic part are described as amphoteric. They exist mainly as ions and can carry both a positive charge on the basic part and a negative charge on the acidic part. Such ions are described as dipolar because they have positive and negative polarity. Having negative and positive poles, such ions are called Zwitter ions. Zwitter ions. This explains the fact that amino acids and proteins can be made to move in an electrical field as when they are separated by electrophoresis. Kwa hiyo utaenda kuona kwamba kwenye methods za separation za proteins, hii sio kwa level nne. So kwa advance amsomi method za separation. Lakini ukija kwa mfano chuo unasoma sasa different method ambazo unaweza ku separate proteins. Electrophoresis inatumia electrical field kwamba amino acid inaweza ka move kwenda kwenye negative charge au kwenda kwenye positive charge. Kwa sababu gani? Inakuwa yenyewe iko charged. So electrophoresis is a modified form of chromatography used to separate charged molecule. Ni modified form ya chromatography ambayo inatumika kuseparate molecules. Hii unaweza kaona maelezo yake kwenye appendix 1 ya BS. Appendix 1 book 2 ya BS. The charge on amino acid can be affected by charges on its environment. For example, making solution more acidic would increase the concentration of hydrogen ions and hence positive charges, and these would tend to cancel out the negative charges on amino acids. Tunachomaanisha ni nini? Let's say to may increase hydrogen ions in the solution. If the hydrogen ions are increased in the solution, these may come to associate with this oxygen. And what will happen is that the negative charges in the amino acid in solution will decrease. And maybe if we increase hydroxyl, these may react with the hydrogen ions. So the positive charge in the solution will increase. The positive charge in the solution will decrease. Why will decrease? Because the positive charge, the hydrogen ions, have been reacted with the hydroxyl ions. So the diagram here shows the illustration of the formation of the Zwitter ion. As you can see yourself that here the hydrogen is released from the oxygen of the carboxylic acid and it goes to join there in the nitrogen. So hydrogen atom dissociate from carboxylic group and associate with the amino group. So the resulting ion looks like this, which is nitrogen with 3 hydrogen and the oxygen is here. So the carboxylic group becomes negatively charged. The amino group becomes positively charged. So this is neutral Zwitter ion form of amino acid. And sometimes if these exceed means the negative charge will increase, the positive charge will increase, and if these exceed means the negative charge will increase. Note, since ammonia, this amino group being a base, possess high affinity to hydrogen ions, hence can attach to the hydrogen ions from acidic group, and the acidic group remain positively charged. The acidic group remain negatively charged. This is not positively, this is negatively charged. So is like how is seen here in this diagram. So in the next video we will go to see about the range of amino acids. So just stay with me, subscribe the channel, and we shall go to see different categories and even classification of amino acids. before we are going to jump on the formation of the peptide bond and the formation of different kinds of proteins. So subscribe the channel and share the video to your friends, and even share the video to different groups so as to learn together with your fellows. Nice studies. Thank you.